Welcome back to my course Industrial the aspects of biochemical engineering. Now today in this lecture I am going to cover a new topic, that is the immobilization of enzymes. In the last couple of lectures I try to concentrate on enzymes, and we discussed the theoretical part of enzymes, that what is the enzymes, and how you can determine the activity of the enzymes, then how substrate enzyme interaction takes place, and and also how you can find out the volume of the reactor for getting a desired amount of substrate, and also we also discuss about the enzymatic inhibition. Now immobilization enzyme has a lot of industrial applications actually, because we know that major drawback of this enzyme is the stability of the enzyme, because we cannot because the enzyme cannot be stable for longer period of time. The immobilization is one of the techniques through which we can improve the stability of the enzyme for a longer period of time. And not only that, because when you talk about any kind of enzymatic reaction, after the reaction is over, the enzymes remain as the impurities in the reaction mixture. So after the reaction is over, you have to remove the enzyme from the reaction mixture. So that is another problem that we have, and that is why that when you use the immobilized enzyme, so you don't have that enzyme in the product, much of the enzyme that is not present in the product. So your purification problem, that will be little bit easier, and not only that, that you can use the enzyme for longer period of time, because in case of normal enzymatic reaction, after the reaction is over, you have to, the enzyme cannot be reused again. But in case of immobilized enzyme, you can reuse the enzyme again and again until its activity declines. So this lecture will be deals with particular this immobilized enzyme process. And first thing that I want to highlight, what do you mean by immobilization? Immobilization defines as the process of confining of cells or enzyme in a solid support. What do you mean by that? Suppose this is a solid support, am I right? And your enzyme that is that immobilized. So when suppose this is a solution and the enzymes are there in the solution, so they are freely moving, am I right? But when it is fixed on a solid matrix, their movement is arrested. They are not moving, they cannot move. So the advantage is that, so suppose you have a column here, you have a small column, and you immobilize the enzyme, you can pass your substrate here and you can get your product in other end. But here, when you pass your substrate and get the product, with the product you will get the enzyme also. So here you will not get much of enzyme. So basically that immobilization defines as you are fixing the enzyme on a solid matrix, and movement of the space is restricted. That is the major thing that we have, and the support or matrix on which the enzymes are immobilized allows the exchange of media containing the substrate and effector and inhibitor molecule. So what I want to tell, that suppose your enzyme is immobilized here and your substrate is here in the bulk, so this is to diffuse here, and then and only then your reaction will take place, and after the product form, the product will come to the bulk. So this is the process that will take place in case of immobilized system. Immobilized enhances the stability of the enzyme. I already discussed this increases the stability of the enzyme. The advantage of the immobilized system, that first is the stability increases, that already we pointed out, can be reused again and again, that also I pointed out. So suppose in a big column, so that you can put your immobilized enzyme here, and here you pass, suppose we want to produce glucose to fructose, the glucose isomerase enzyme, glucose isomerase. So, we immobilize that, immobilize the glucose isomerase here, he will get the fructose. So, so you know that this you can operate it continuously, and until unless your reaction declines. Okay, and products are free from enzymes and can be used continuous product formation, suitable for industrial and medical use. How it is medically used? Because we know that due to some genetic disorder, that sometimes some of us are unable to produce some kind of desired enzyme. So, that enzyme we shall have to take from outside for our survival. Now, if you take the enzymes in the form of tablets, then you have to take it you have to take lot of tablets. Now, suppose some enzymes, if you keep it immobilized and put it in the bloodstream, so you can use that enzyme that for a longer period of time. You don't have to take the enzyme again and again. So, this is one of the important application we have in the medical sector, medicinal sector, and the minimize the effluent disposal problem, since it doesn't have your enzyme in the product. So, your disposal problem also will be reduced. Now, application of immobilized system, that examples, if you look at the industrial application, we have antibiotics, beverage, and the amino acid. Because I can give a very typical example, that chill proof beer. I don't know whether you have heard that, chill proof beer. What do you mean by chill proof beer? The beer is usually served under chill condition, and we know the beer is kind of beverage that contains lot of proteins. Now, since when it served at low temperature, there is every possibility that if there is a protein, bigger molecular protein, it may precipitate it out. Now, if it precipitated out, it will give some kind of hazy in the solution. That is undesirable. So, this is in the lot of application. This is one of the application that we have in the beverage industry. Then medicinal application, I told you, treatment and diagnosis. Diagnosis means that suppose particularly during the athletics competition, that if we want to find out that whether the athlete they have taken any kind of stimulant, and so we can have a sensor, that biosensor. If we have, we can form the biosensor, we can easily detect that whether they have taken any kind of stimulant. Drug delivery system, this can be used. Food industry, it is used for the production of high fructose corn syrup. This is mostly used in the confectionery industry. And waste treatment process and sewage and industrial, particularly trickling filter, or that is largely used in the industry, that for the removal of the soluble organics present in the wastewater. Now, hypoxidase, the breaches the white in white bread, and peroxidase, the paper manufacturing industry. Hydrolase, we have alpha beta amylase, that is used in brewing industry. Cellulose, wine making, and the glucoamylase, the starch processing. Penicillin amidase, that is the antibiotics industry. Keratinase, leather manufacturing industry. Lyase, fumarate hydratase, that the malic acid, and isomerase, glucose isomerase for fructose syrup production. These are the different industrial applications we have. Now, as I pointed out, that one important application that we have is the biosensor. Now, question comes, how biosensor they are produced. Now, here in this picture, you will find that two part is there. One is this is bioreceptor, and this we call molecular recognition unit, and this is called transducer. Because so it divided into two different parts. One is molecular recognition unit, another is transducer. The molecular recognition unit, that main part was they recognize the molecules. As for example, suppose the glucose oxidase we have. Now glucose oxidase, in presence of glucose, immediately they will react and produce degrade to gluconolactone. So that reaction is there. And that reaction, that chemical change, we can convert it to the electrical signal, that is with the help of transducer. And this is mostly, this may be of different types. The mostly we have two different types. One is potentiometric, another is amperometric metric. The potentiometric means with respect to change of voltage, and amperometric means with change of current, that we can easily find out what is the change that takes place, because and that we correlate with the concentration. I can give a very simple example. Suppose this is microvolt and this is the concentration of substrate, maybe glucose that we have. So it is like this. So the major problem is that after this concentration, this attains the plateau. That is the major problem. That means it is effective up to this concentration. After this concentration, it is not effective. So that is the major problem when we use as a biosensor. Your concentration of the sample, concentration of the substrate should be as low as possible. So if the concentration is high, then you still have to dilute significantly to have this. But the advantage is that you can analyze n number of sample, and instantaneously you can do that. It will not take any time. But if you go for any analytical techniques, you have to prepare the sample, you have to add different chemicals, then you have to put in the spectrophotometer. It takes a lot of time. But this is the major the frequency of analysis that increases drastically. So this is how it is done. This is the bioreceptor, and this is transducer, and this is how we can detect that that in a detector we can detect that how much we correlate, that how much that the first you can see that here that the biomolecules, they are immobilized on the surface. I can give the example that the enzymes are immobilized on the surface, and enzymes are very specific with respect to substrate. When they react, it is converted into different compounds, and when this reaction takes place, it is converted into electrical signal, and this electrical signal we recorded in our recorder. Now, classification of solid matrix, the solid matrix that is used for immobilization purpose, because the different type of solid matrix we consider. One is natural polymer, then synthetic polymer, then inorganic. We have natural minerals and processed minerals. Now, what is the natural polymers? We have polysaccharides like cellulose, dextrin, agar, agarose, chitin, and alginate. Protein, we have collagen largely used, particularly I work with IIT Delhi, and we use the collagen membrane for the immobilization of glucose isomerase enzyme, albumin, and also that carbon. The synthetic polymer, we have polystyrene, and other polymer we have polyacrylate, methacrylate, the polyacrylamide, the polyamide, and vinyl and allyl polymers. Natural minerals are bentonite and silica. Then processed minerals are glass, metal, control pore metal oxide. So this is the different examples of the solid matrix. So when we talk about any kind of solid carrier, broadly it can be divided into two types. One is called non-porous. One is called non-porous, and another is porous. That means that suppose that this is solid matrix, so it doesn't have any pore, but this solid matrix may be having this kind of pore. So that this is how pores inside the pore, that that some pore is there inside the solid matrix. So this is non-porous, this is porous. So in porous also, this pore may be of two types. One is control pore and there is broad pores distribution. That means when the pore size is uniform, suppose your pore size is uniform like this, then this is a control pore. So that is obviously that will be very costly, and broad pore distribution means pores size are different. Some size is bigger, some size is smaller, like this. Inside this solid matrix we have that different size of these pores. Now non-porous, what is the advantage? The diffusion effect is minimal, because it is very obvious, because suppose here this is solid matrix and enzymes are immobilized on the surface. So when your substrate is going, that it can freely interact with the enzymes. This is the enzyme. This freely interact with the enzymes. The diffusion effects are minimal, and large substrate in solution can be reacted with minor difficulties. Disadvantage is the lower surface area, because since the surface is smooth, that surface area will be very low. Now porous has lot of advantages, because let me tell you, the first is the high surface area. This is the major advantage is so since you have porous surface, so with that porous surface, that that we have as compared to smooth surface, so obviously that this porous surface has lot of area. So more enzyme, if you have more surface area, more enzyme will be immobilized on the surface. The internal surface bonding protect the enzyme. Now suppose this is how your solid matrix. Now if your enzymes are here, it gives some kind of protection, because it is embedded, or when we talk about the suppose encapsulated the enzyme. So it has some enclave, that can protect the enzyme. The internal surface bonding protect the enzyme from the turbulence or external environment, and a charged surface opposite that of the surface may enhance the surface enzyme substrate interplay, attracting the substrate. Suppose here we have, if we have the positive charge and your substrate has a negative charge, obviously the interaction between the enzymes interaction with the substrate with the enzymes will be more, because your matrix charge is opposite to that of the substrate. And large enzymes or cell loading possibility, since pore size is more, so it is obvious that loading also will be more. Now there is a lot of disadvantage also there. Large enzyme and substrate molecules cannot be penetrated in the smaller pore. But there is a rule of thumb, I can tell you, that I shall show you at the end. There is the enzyme called catalase enzyme. What is the function of the catalase enzyme? It degrades the hydrogen peroxide to water and oxygen. Am I right? And this is largely present in the aerobes, because aerobic organisms. Now, in case of anaerobic organism, particularly faculty that is obligatory anaerobe, they don't have this catalase enzyme. And since they don't have this catalase enzyme, so when oxygen goes to their system, it produces hydrogen peroxide or superoxide, and this superoxide has oxidizing property. So since they have oxidizing property, they will change the characteristics of the biomolecules. And if they change the characteristics of the biomolecule, then the activity of the enzyme or activity of the cells or enzyme will be lost. So this is the major problem that we have with this sports. So large molecules cannot penetrate this. So what problem we have is, let me tell you, that when you the rule of the thumb is that the size of the pore should be double the size of the enzyme. Suppose the size of the enzyme is 84 angstrom unit. So, it should be 2 into 84 angstrom unit, that is 168 angstrom unit should be the pore size. But in case of catalyst, though it is round about this 84 angstrom, but we find that if you plot like this activity of the enzymes with this, we will find to keep on increasing with the pore size, and this pore size is something similar to 364 or something like this. Now, question comes why? Because it required some now I hope I told a little bit different that this is I am talking about the glucose oxidase, the glucose oxidase enzyme. Now, in case of glucose oxidase enzyme, when it acts on glucose, it produces hydrogen peroxide, and this hydrogen peroxide should be degraded by the catalase enzyme. Now, hydrogen peroxide has the pore size 84 angstrom unit. So, when we plot the activity of the glucose oxidase, we find it is keep on increasing, but maybe at 384 or 87 the angstrom we get the we get the maximum activity. But the reason is that this catalase has the size much higher than this, and we find that this size is related to the size of the catalase enzyme. So, large molecules or substrate cannot penetrate. This is the major problem that we use the porous solid matrix. Now, if the pore has same charge as substrate, then also interaction will be less. Broad pore, if your pore size is more, then also this is undesirable. That there is every possibility that their enzyme substrate interaction will be very less. Entrap or encapsulation are severely limited by the diffusional effect. So, this is the diffusional effect, means if a pore is there, then substrate has to enter into the pore. So, naturally your diffusional problem in this case will be very stringent. The microbial resistant property, that is very important. That is, if you look at the carrier which are rich in carbon, such as starch, or rich in carbon, nitrogen, such as protein, are good nutrient to the microbes. Now, if these molecules are present as a solid matrix, then there is every possibility that your microorganism can grow and spoil the solid matrix. If the carrier is attached with the microbe, the enzyme then released into the solution. Now, inorganic oxide like silica, alumina are resistant to the microbial attack. Organic material such as fluorocarbons, polypropylene, etc. are resistant to the microbial. So, microbial resistance property of the solid matrix, that is also plays very important role when we select this what should be the solid matrix we should use. Now, what the question comes, what should be the characteristics, desirable characteristics of the solid matrix? First, it should be that chemical durability. What do you mean by that? That suppose you want to carry out a reaction A to B. The B is a new product, A is a chemical product. Now, it should withstand both the A and B. Otherwise, if degraded, then that is undesirable. Then high available surface, I already discussed just now. More surface area, more there will be enzyme that will entrap, and if more enzyme is there, more it will interact with the substrate and give the product. Mechanical strength and dimensional stability, this is very important, because as you know that when suppose if there is a solution, and when you do the immobilization, that what you do, that in the solid matrix your enzymes are immobilized. Now, what is the characteristics of the solid matrix? They will settle down. Am I right? So, if you want to have more interaction with the substrate, you should have mechanical stirrer, and if there is a mechanical stirrer, so your solid matrix should withstand this mechanical force, mechanical strength and dimensional stability. Then microbial resistance, this is obviously to avoid the destruction both the enzyme and carrier, and surface and pore charge with respect to the surface. Pore charge should be, if preferably should be opposite to the substrate, so that more enzyme substrate interaction take place. thermostability, particularly we talk about the alpha amylase enzyme that usually act at the high temperature as high as 60 degrees centigrade. So if we select any kind of solid matrix for alpha amylase enzyme is to withstand that high temperature. The carrier shape and particle size that plays very important role. We know the spherical size, the shape it has more surface area as compared to other solid material. Now regeneration of the carrier that plays very important role. Suppose we want to use any kind of solid matrix, maybe it is very cheap, but we might be using for single time. But so yeah, again when you but some of the solid matrix, I can give the example of platinum foil that is very expensive. Platinum is costlier than gold, but platinum foil you can reuse again and again. Then for longer period of time, after when your enzymes are inactivated, you take it out and put it in at 600 degrees centigrade for 2-3 minutes. All the organic material will burn out. Again you can use it is for immobilization purpose. So regeneration of that solid matrix, though the initial investment is very high, but you can reuse again and again. So regeneration also plays very important role. Now carrier regeneration, what I was talking about, this is life cycle of the solid matrix is very important. That how long you are going to use. The organic material causes the disposal problem, that is very important. But inorganic carrier can be regenerated and relatively simple we can regenerate with the help of simple pyrolysis process. I was talking about by simple heating we can do that. Now this is the properties suggested carrier applications that stick. This is silicon rubber and nylon methylylate, that is medicinal interface with the blood and tissues. Do not initiate the clotting reaction, little or no immune response evoke. The collagen, this is another the cellulose acetate that is used. This is for polymer hydrolysis, collection of product-free substrate. Then control for glass that is used, continuous plug flow at pH below 7, dimensional stability, low pressure drop and duration durable below pH 7. Control port alumina, continuous plug flow reactor 5 to 11, dimension stability, low pressure drop and durable above pH 5. Control port titania, this is continuous plug flow reactor. So these are the different properties of some carriers that has been reported. Now optimizing the carrier, that is the port diameter, as I pointed out, that important consideration. Major axis of the enzyme, the unit cell should be greater than the major dimension of the substrate. Then the port diameter carrier should be chosen with respect to enzyme. I was giving the example of glucose oxidase, that glucose oxidase enzyme. So so in this following example, that we can find out that here we have this is the glucose oxidase enzyme activity. And this is exactly what I am saying, that this is around 384 or something like the angstrom unit. And if you look at the size of this is 84 angstrom. So this should be into by 2, it should be 168. Am I right? Angstrom. But it is about 384. So this is due to the reason is that glucose oxidase cannot work without the presence of catalase enzyme. Because why without catalase enzyme it cannot work? Because catalase enzyme, the one of the product with the glucose oxidase is the hydrogen peroxide, and catalase enzyme that degrade the hydrogen peroxide to water and oxygen. So that is why otherwise if you don't use the catalyst with the glucose oxidase, then the accumulation of hydrogen peroxide will take place, and your activity of the enzyme will be lost. So that is why we find in case of glucose oxidase enzyme about 384 or 380 angstrom will be most suitable for the immobilization of the enzymes. So so in conclusion, what I want to tell, that immobilization is a very important techniques through which we can use we can make proper use of the enzyme, because it has several advantages. It has some disadvantages also. The major advantages is that you can reuse the enzyme again and again for longer period of time. Your product is free from enzyme, so your purification problem will be little bit easier. Your disposal problem also very less. But the problem is that when you immobilize, that means you are fixing the cell on the solid matrix. The diffusion problem is the major problem. Then a question is the solid matrix, because as soon as you immobilize that, suppose your enzyme is soluble, as soon as you immobilize that, it will become insoluble. So you are switching over from a homogeneous system to the heterogeneous system, and heterogeneous system major problem we told you, that is the diffusion problem. The diffusion problem we have to take into account, and characteristics of a solid matrix. I try to discuss the characteristics of the solid matrix is that it should withstand chemically stable. It should withstand it should have good mechanical power and regeneration characteristics, and surface area should be as high as possible. So the different characteristics we have, and we have seen that solid matrix may be of two types. One is pore, and there are non-porous. So in porous, we have a porous solid matrix having greater surface area. So there is a possibility of more immobilized enzymes. So if more enzymes are immobilized, more will be the rate of reaction. So that is desirable. So that is all for today. That next class I shall give some more information on the immobilized enzyme. Thank you.